Our next speaker is Dr. Hannah Carey. Dr. Carey is professor in the Department of Comparative Biosciences at the University of Wisconsin School of Veterinary Medicine. She is a graduate of UC Davis and done postdoc work at University of Nevada and Ohio State University, focusing on hibernation biology, translation of hibernation, effect on GI and liver physiology. Dr. Carey is past president of the American Physiological Society and past chair of the Nutrition and Obesity section of the American Gastroenterological Association, amongst many other appointments. Annual cycles of extreme dietary change shape, gut microbiota, and their hibernator host. Dr. Carey.
Thank you very much. I'm really pleased to be here, and I've enjoyed the talk this morning. Angela, that was a nice one as well. Some interesting points that are a nice lead into another model that takes us up definitely a phylogenetic level here. So this is an animal that I think could be perhaps the most dramatic example of phenotypic change in the mammals that occurs on a regular basis throughout the lifetime of the animals. And this is seasonal hibernation. In response to periods where energy availability is low in the environment and energy demands are high, typically winter, these animals go into a dormant state and forego many of the normal physiological, behavioral, and metabolic processes in favor of a new mode of living for up to, in some species, almost a year at a time. So let me tell you a little bit about the model and why we're thinking more and more this is an interesting model for understanding host microbe, in this case host gut microbe interactions because of this extreme lifestyle they undergo. The 13-line ground squirrel, which is a particular really good hibernator that we study in the lab, is characterized by the typical response. Animals are lean in the springtime after the last hibernation season, and they begin eating quite rapidly, voraciously, and fatten to pretty much obese levels by the end of the summer, early fall, before they hit into the next hibernation season. And then they voluntarily cease feeding. So they've got some wonderful energy-sensing gut-to-brain appetite signals that we'd love to know more about that tells them it's time to stop and to exist in a different metabolic mode. Along with the feeding pattern and the body weight cycles is a remarkable pattern of energetics and body temperature regulation in which the animals are essentially homeothermic from spring through early fall, and then they become heterothermic. And by heterothermic, I mean what's shown here. These lines right here is an actual body temperature trace taken from one of our ground squirrels with a telemeter implanted. These are in the laboratory cold room held at the animal housing cold room held at 4 degrees Celsius. So once the hibernation season begins, the animals undergo bouts of torpor, which is a very depressed metabolic state, and body temperatures actually in some hibernators, like the Arctic ground squirrel, have been clocked in below zero, -2, -3 degrees Celsius. Our squirrels tend to hover close to zero, but usually don't go below. So these bouts of torpor are energetically wonderful because they greatly reduce the need to take in new nutrients. But you'll notice the hibernation season is a very dynamic period, and these torpor bouts are punctuated by arousal to normothermic conditions. This happens underground without any environmental cues. It happens in a laboratory cold room. The animals are normally held at about 4 degrees ambient temperature. We take away food and water. Once they start their torpor cycles, we turn off the lights, and it's just a brief few minutes each day that we do a daily check. And they'll undergo these arousal cycles without any outside input. So there's some metabolic signal, presumably, or I really should say a metabolic, what we think then is transduced to a neural signal that's stimulating these arousals. But I point this out because although this is a period of very depressed metabolic state, both for the squirrels and for any symbionts in their bodies, there are accumulatively a number of hours spent at high body temperature when the animals are metabolizing as well as microbes are metabolizing. So again, this is a period with no feeding, no dietary input, and the animals, the hosts, rely primarily on all these stored lipids, the fat they've been storing for fuel. So after many years of looking at what this means in terms of the gut, the GI tract for the ground squirrels, we've more recently turned our attention to the other organisms that inhabit this environment within the animal. Let me point out by telling you the short end of a lot of work, in this case structurally and later functionally, that the nice typical small intestinal mucosa of a mammal becomes atrophic during the wintertime. The normal crypt-villus architecture is still present, but there's a lot less tissue there, which makes good sense. The animals aren't eating. They don't need a robust mucosa to finish digestion and undergo absorption of the food. So the normal digestive processes typical in any mammal, and the summer ground squirrel, is curtailed. Food processing and absorptive functions are largely absent. And in fact, this is why most people in hibernation biology haven't paid much attention to the gut, except for my lab plodding away pretty much alone for many years. But when you realize what has to continue during hibernation in terms of the gut is this continued need for a good barrier function and a mutualistic, maintaining a good relationship with the resident gut microbes. So that's what we've been exploring in recent years. This is micrographs from the small intestine, and this is a picture of the cecum, where we've been doing a lot of our microbial sampling and paying more attention. This is taken from a hibernating ground squirrel and points out that, as I said, no diet is taken in for the animal. They have fat to live off of. What about microbes? Microbes, many of them do make use of dietary components that escape digestion. In the wintertime, there is no diet. But there are endogenous host substrates that can be utilized at least by some microbes that have the metabolic, the enzymatic machinery. And mucin, the mucus layer, is certainly one of them, shown here in this kind of aqua color coming from the goblet cells. This is a schematic of that life cycle, and it shows you how we're doing the sampling to understand what's happening to the microbial communities during hibernation, or the whole annual cycle, and in turn, ultimately, what's happening to the hosts that might reflect the need to maintain the mutualism with the microbes. So first few slides is about the microbial side, and here's what we do. We capture pregnant females in the wild. They give birth in-house in the animal facility, so we have full control of the diet of the pups that are born. In the summertime, the mothers that produce the pups are sampled, so we get an idea of the mother's maternal microbiota, for which we can compare the young. And then we start sampling the young are randomized among these different seasonal groups, and we have a summer group that sends samples. The rest of the animals are put into that cold room, and food and water are taken away. Hibernation begins. And then we have what we call the early winter group, animals that have been hibernating for just 1 month. They haven't had food, but 1 month of both these torpor and arousal bouts, and we sample in both of them to know if the state of torpor is making a difference in terms of the microbial community. Then we have our late winter group, 4 to 5 months of hibernation. That's 4 to 5 months with no food at all. Same two metabolic states are sampled, and then we take whatever the remaining squirrels are left, put them back into the warm room, give them back food, water, normal light-dark cycle, nice warm environment for 2 weeks, and then those are sampled. So that's our spring group. And in the microbial studies I'll mention, we've been looking at the cecum, high density of microbes there. DNA is extracted either from fecal contents or the tissue. The fecal content data that's now recently been published, this work was done in collaboration with Rob Knight's group and his graduate student Tony Walters at CU Boulder, and that was the luminal microbiota, so content. But we realize, of course, content is very different in the summer versus the winter. In the winter, the content in a hibernating squirrel, what's in the lumen, is all endogenous fluff material, mucus, fluff cells, pancreatic biliary secretions. So we realize we should also pay close attention to the mucosa-associated microbiota because of this difference in the luminal content and because of the growing realization that the mucosal population may be quite different from the luminal population, may not, but it has even a closer, more intimate association with the host. And this work that's just been beginning is being done with Garrett Sewin and his group at the University of Wisconsin, Department of Bacteriology. The luminal microbiota data are shown here in a principal coordinates analysis, give us an idea about the phylogenetic relationships among these samples. And each of these individual points is a ground squirrel, either a mother or some of the offspring. And when we look at how these cluster, it drops out very nicely. Maternal influence seemed to have no effect. Didn't matter who your mother was. It also didn't matter what state you were in when you were sampled, but the time in the season. This time without food seems to make a big difference. So the active season here, we have the mothers are interspersed with the summer, and the spring squirrels, which are the most diverse. That's the green squares here. And this is our active group. And then the winter animals. The blue is the late winter animals. Red is early winter. And what about the torpor versus arousal? Did that make a difference? Turns out no. The circled dots are all the animals in the winter season that happen to be torpid. Well, we specifically knew they were torpid when we sampled them, and they're interspersed throughout these two groups. So what the microbiota is at that point in the season is what seems to make a difference. This is some alpha diversity measurements of phylogenetic diversity and species richness, or numbers of observed OTUs. And late winter is the lower, is the least in both of these measures. So that is the most contracted population all the way by late winter. And somewhat surprising, the highest diversity and the greatest number of OTUs was in the springtime. Maybe upon further thought, it's not surprising. This is just 2 weeks. There's only 2 weeks that separate spring and late winter, and those are the two extremes. So there's a bloom, if you will, of lots of species shortly after emergence from hibernation. And then things settle down. Here's the summers in the purple. Things then settle down from spring a few months later into the summer, when there's lots of feeding going on and lots of animals have been high metabolic rate for a long time. So the spring tends to be the most diverse period here. Who are some of these organisms? This is, again, the luminal microbiota. And what we see most dramatically is a shift in the proportion of sequences assigned to the firmicutes versus the Bacteroidetes. You have the firmicutes in the blue. They contract during the winter time, especially in late winter, and start to return in the spring. There's an increase in the Bacteroidetes. Something somewhat surprising, kind of interesting, was the Verrucomicrobia, which are a very interesting group of organisms. And the verrucos altogether represent a fairly high percentage for the other mammalian microbiotas that I've been looking at in the literature. They're well represented in the ground squirrel in general, but they really come up during the winter time. And although they start to contract again in the spring after those 2 weeks, there's still carryover there. And this is there's a lot of interest in this group, as Angela mentioned. They have been described to be mucin dedicated mucin degraders. So this may go well along with the idea that one of the big substrates in the wintertime, one of the only substrates available, is the mucus glycoproteins, the mucus glycans available. But there's a lot of interest, too, in the relationship between the Verrucomicrobia and signaling and host responses at the mucosal level. So this might play a role in some of the things I'll tell you coming up here. That was at the phylum level, the family level. Similar idea, just broken down a little bit more. Among the firmicutes, seems to be particularly the Lactospiraceae that really go down in relative abundance. The lactobacilli just drop out completely. There's lactobacilli in the active seasons, and these purple bars just disappear. So so here we have some taxa benefit from the altered microbial community, from the altered environment during hibernation, like the Verrucomicrobia, some of the Bacteroidetes. Others are disadvantaged, if you will, by this change, and that could be the lactobacilli and some of the other firmicutes. Just a little bit about some of the very recent data about the mucosa-associated microbiota. There are some general patterns that are the same. The firmicutes contract, Bacteroidetes come up. The vertical microbia are somewhat surprising to me, didn't constitute such a high a percentage in this particular population, but they also undergo a similar shift with an increase in the winter time. And at the family level, again, we can see some of these changes. And there are some interesting, less abundant, but significantly changing species that we're interested in, particularly in this late winter, which is the most extreme state of a gut that hasn't seen food for long periods of time, that we're exploring in more detail. Okay, let's turn to the host side. This is more difficult than just describing what species of bacteria are there because, well, we can describe what may be changing in the animals that we think are related to this relationship, but it's harder to nail it down. We can't make germ-free ground squirrels, so we're doing our best with playing around with antibiotic regimes now, which have their problems as well. But let me tell you what we've found that we think might be a reflection of the host response to this changing winter environment and perhaps its relationship with the microbes. First of all, importantly, the gut is leakier during hibernation. We know this from way back years ago when we were doing a lot of lifting chamber studies that are in vitro. We put up in vitro pieces of a tissue and looked at electrical passive permeability through the paracellular space, and this is in small intestinal tissue. So conductance is consistently higher. It's leakier, at least in terms of that paracellular level. But more recently, we asked, well, what about macromolecular transport on a whole animal level? So we used the 50-dextran absorption method and have found that indeed there is more a leakier gut in terms of macromolecular transport. So this is one of the risks, if you will, of going without food for a long period of time. This has been seen in other models. The absence of enteral nutrition can increase intestinal permeability and put an organism at risk, if you will, for translocation of bacterial products across the epithelium and potential effects there on the underlying immune system and the rest of the body. So the gut is leakier. There is a pretty significant remodeling of the intestinal immune system during hibernation, perhaps in response to this leakier gut and or like moderate levels of bacterial product movement across the epithelium. So here we see summer on the left, summer squirrels, hibernators on the right, and these boxes blown up underneath here. And pretty obvious just looking histologically, we see an increase in lamina propria lymphocytes here, and this is, again, small intestinal sections, and really a pretty noticeable increase in intraepithelial lymphocytes. This lymphocyte population that's sort of at the first line of defense, often lodged up way between the epithelial cells, close to the lumen where the bacteria would be. And that's been quantified through work done by a nice graduate student and now assistant professor in our lab who we're collaborating with again. Especially in the torpid animals, these intraepithelial cells are high, and even though they go down during the aroused state, they're greater than the summertime. Lamina propria lymphocytes also increase. In the lamina propria, we see an increase in the proportion of B cells, and actually that somewhat goes along with our finding that there's increased amounts, and this is tissue level, not in the lumen itself, but of IgA, secretory IgA, an important master regulator of both protection against bacterial adherence and invasion across the mucosa, but it appears IgA may be playing a very complex role in regulating the relationship of the microbial community to the host. We also see a number of interesting cytokine changes during hibernation. Some, quote, pro-inflammatory cytokines, TNF-alpha, interferon gamma, also IL-4 is elevated, and a fairly profound increase in IL-10, prototype or classic anti-inflammatory cytokine that's elevated not just during the arousal process, but throughout the winter time. Higher levels of IL-10 in the mucosa. Some very new data, too, are giving us some idea about perhaps changes in bacterial sensing and response in hibernation. From summertime to early winter and then late winter, we're seeing an increase in TLR5, the flagellin receptor. This is in cecum tissue, along in this collaboration with Courtney Kurtz and her students at UW Oshkosh. And this data that just came out, preliminary data suggesting that ileal registry gamma levels are also increased during the winter, and this again an antibacterial leptin that is produced by epithelial cells as well as intraepithelial lymphocytes. So we think that the intestine during hibernation is not involved in food absorption and digestion anymore, but it does have an important role in maintaining that barrier. Modifications to the intestinal immune system may be important to keep that good mutualistic relationship with the microbiota. What other things could help besides the immune system? In some work we did with Jerry Turner and Chris Weber, we've been looking to see if there are any changes in tight junction proteins right here between these enterocytes, that all-important paracellular passageway. And indeed, there's some very interesting evidence that Chris Weber got through some immunohistochemical staining for occludin, one of these tight junction proteins. Here's small intestine and cecum, summer, winter, and you can see this really nice punctate staining up at the point of the tight junction for occludin in the hibernators, small intestine and cecum compared to the summer months. So this may be a compensatory response to try to limit that level of enhanced permeability that accompanies winter fasting. So to summarize here, we're starting to put together the pieces of the cycle here of the summertime to early winter transition, changes in the microbiota, going along with the animal's overall cycle. I haven't talked about short-chain fatty acids, but I'll just say that, not surprisingly, total short-chain fatty acid concentrations in the cecum are greatly reduced, although still present during hibernation. There's no substrate for microbes to ferment in terms of diet, but they're fermenting endogenous host glycans like the mucus. There's an interesting shift in the molar proportion of the SCFAs that do remain, with increased relative amounts of acetate and a real drop-off in butyrate levels. Perhaps this is drop-off in butyrate related to the atrophy that occurs, because butyrate is a preferred fuel source for proliferation of epithelial cells. So we have the feeding to fasting transition and some of these features that I just mentioned. And there are interesting questions to continue on through here, ways that we have to be clever to get our answers with this model. One is whether there is a core hibernator microbiome, and this we're doing with collaborators here in Wisconsin, Colorado, and up in the Arctic tundra, looking at the Arctic ground squirrel. And these are diverse ground squirrel lineages, all good hibernators, but they evolved in quite different habitats. And we're looking to see if, at least at the level of OTUs, at the taxonomic level, if these animals all converge in because of the physiology to a core microbiota. And ultimately, we'd all like to get to the level of metagenomics and metatranscriptomics and get beyond the species and look at the actual functional output of these hibernator microbiomes. Other interesting questions at the microbial side: what's the role of dormancy? Actually, some of the literature I read preparing for this meeting reminded me that there's a world of interesting questions in terms of what's happening to these microbes that are going close to 0 degrees. Are there some that are in a dormant, resting, or sporulated state? These are things we just don't know, but it's fascinating to think about. We're delving deeper into the immune changes and trying to get more at the signaling mechanisms between host and microbe in hibernation. We're also interested in whether and how these microbes contribute to energetics, nutrition. It's been long suggested in the literature of hibernation that nitrogen protein conservation, which is absolutely important in hibernation, is helped by nitrogen recycling due to gut microbes and the urease activity. But there's very little data that backs that up, so that would be something we're quite interested in looking at. And finally, other work we've done in the lab suggests that the intestine during hibernation, as well as the liver and other organs, become quite stress tolerant during the wintertime. These animals are tougher in the winter than they are in the summer, and certainly compared to a typical lab rat. And we know this, for example, from ischemia reperfusion injury studies, that the intestine is much able to bear an insult. I never thought about it until we started working on this, but there may be some role for the microbes in helping to provide that stress tolerance during the wintertime. So this is a beautifully co-evolved relationship between these animals and their microbes over time. And this animal is my last slide here, of course, it makes us very excited about translation possibly of some of these principles to humans. Does anyone know what this is? Zoologists in the audience, a lemur, that's right. This is the fat-tailed dwarf lemur. It is a primate, and we now know that the primate lineage has the ability to hibernate, making many of these pathways, both from the host side and perhaps this symbiotic side, even that much closer to us. So with that, I'll just put up some of the people that contributed, the many collaborators that helped with the studies, and thank you for your attention.
Thank you very much. It's up for questions, starting with forum members, Margaret.
Yeah, that was a wonderful talk. I have just a couple of questions. Well, I have a bazillion, but I'll just ask a couple. So one of the things I'm wondering is, since there's a lot of information, actually I'm first going to ask just for a clarification. You had no maternal effect? Does the ground squirrel not have coprophagy, or are they coprophagous?
Yeah, good question. Coprophagy has not been observed in these animals.
Okay, so that can't explain.
We don't prevent that, but we think it's not.
Okay. So the other more deeper question I had was about the gut-brain axis, and I'm wondering, this interesting arousal period and how that might work in microorganisms and whatnot. Is there any information on feedback at this point between the resident microbiota and the brain of the animal? And the second thing is that have the microbiologists looked at the behavior of the microbes under culture conditions to see what happens to those populations, if you can, in a chemostat or something, when you do this?
Right, good question. The first question, absolutely not. Nothing's been done. Several years ago, we started doing some metabolomic analysis, looking at tissue and blood in particular. And at the time, I wasn't thinking so much about the microbes, but I'm going back to those data and thinking about more to look to see if we can find signatures of microbes in the blood coincident with some of these changes that might be candidates for ones that might cross the blood-brain barrier, affect the neural processes that lead to arousal. But I think that's a fascinating possibility. One thing we have seen in terms of metabolite change, and it could have nothing to do with microbes, is a big surge in pantothenic acid, a vitamin, during the torpor cycle compared to the summertime. So the possibility that the microbes may be giving back some vitamins during this time when none are taken in for months and months it's out there, and so I think it's another one. The other question was about the microbes. When I've looked in the literature, and just, I'm not a microbiologist, but I haven't found enough really good evidence to give me an idea about what these kinds of cycles will do. And so I really need to team up with a microbiologist and think about starting to put together some experiments and ask what's happening with the cycle.
Go ahead.
You suggested, Dr. Zazoff, could you please use the microphone?
You suggested that some fraction of the animals that you study don't quite make it. Did you not? Or you said those that survive?
Oh,
what is
those
the,
that
what is
survive
the
on sampling machines.
But in general, what is the mortality?
Yeah, what's the mortality? Good question. Every so often, each year, especially if they're wild-caught animals, we do have some die. Natural causes. When we get them from the wild.
What percent?
1%.
Why did they die?
Well, again, for the ones from the wild, some of them just die as if they were in torpor. It may be during arousal. They live a few years. I don't know. I will tell you one thing, though, that's very interesting, and that is that before, when we first started getting pregnant females and having them give birth in the lab to pups and raising them, we let those babies eat ad lib. I mean, it's a young animal, you let it eat ad lib. those animals got extremely, way more obese by the end of the summer than our normal wild-caught juveniles did. And several of those died in hibernation that first winter with abundant fat reserves. And the pathologist looked at them and said, there's something odd with this fat. And that's all we knew. Now all of our young animals right at weaning are on diets. That's because these animals, I think, are primed as juveniles to gain weight. In the wild, you're running around from predators. You're not sitting in a cage. So in a way, it's an interesting model for juvenile diabetes or obesity, I think. This is not what these animals are supposed to do. They get extremely obese, and it's not healthy for them. So that's a little anecdote there. Okay.
Thank you, Dr. Carey. And again, we're going to have discussion at the end after our last speaker for the session.